Thank guys. They always happy to find the time to be here, and I'm glad to see that Ian is here, and many other very faces that I know. So great to see everybody.
Fantastic. Great. And Peck, we got to say hi to Peck.
Where's Peck?
The day's not complete until we say hello to Peck.
Hey, Peck.
Hi. And I have to say hi to Ramesh, my friend Ramesh, who is joining us from oil and gas, but always interested, aren't you, Ramesh?
Hi, everyone. Yeah, amazing topics and a great community. So thank you for the invite.
Yeah, you're welcome. You're going to have to diversify your sea out of oil and gas, Ramesh.
Chemical engineering.
Well, there you go. That's it.
Tony and Graham, I'm here now.
Hi, Anna. Do you want to just say hi to the group? Good to see you. Good to hear you. Anyway.
Yeah, I mean the thing is, I'm in the car, so I just did a lot of illegal stuff just before. I mean, downloading Zoom and stuff while driving.
You did
Don't do
illegal
that.
stuff.
Yes. So what I'll do is I will park in about 5 minutes, and then I'll join again with camera on from my computer. But I just I didn't want to miss all the introductions.
Be safe, Anna. Yeah, absolutely. A quick hello from Emma. Hi, Emma.
Hi, everyone.
Good to see you.
Nice to see everyone.
Do you want to just tell us who you are, what you do?
Oh, yeah, that would probably be helpful, wouldn't it? Yeah. It's a bit too familiar. I'm Emma Thorpe, Chief Growth Officer of RBW Consulting. We're a life science and technology search firm, and I predominantly work with small to medium-sized biotechs and health tech companies.
Yeah,
terrific.
And Peck, you didn't actually say what you did. I know you said hello. We got excited, didn't we? Do you want to just mention your company as well, Peck?
Okay. Hi, I'm down in the Bay Area. I'm CEO of a company called Oranza, and we are a AI-driven biotech company and really interested in the psychedelics field. I've actually spoken to Chuck, before as well. So really excited about this session.
Great.
Yeah, good to see you again, Peck.
Yeah, good to see you.
And that's Tyler, isn't it? So Tyler, it's I know you're new to this event, but do you want to just say a quick hello and who you are and what you do?
Yeah, of course. Thanks, Graham. This is actually my second session, and I'm glad to be here and listen to the conversation today. I think, like Ian, am a CEO, co-founder of a stealth psychedelic company, working with Oranza and Peck on that project. So, look forward to sharing more in the near future. I'm based in Vancouver. I have a background in molecular epidemiology, and formerly co-founded and led a company called Sequence Bio, which is a population genetics company based in Newfoundland, utilizing that unique genetic population for computational drug discovery.
Fantastic. Thanks a lot, Tyler. Great to have you all here. Tony, would you like to kick it off?
First of all, I'm going to say a massive warm welcome to our Coffee Buddies events. I think this is probably about 130 we've now done, Graham, and we've got more in the pipeline. So you won't be bored during this period. And we've got a lot of fantastic speakers and a lot of things going on. And the ethos of Coffee Buddies is highly inclusive. You never know when we might bring you in. We like to entertain, inform, and engage. And that's what hopefully makes us different from the average webinar, and we certainly don't want to be that. We don't want to be the average webinar. And today we also specialize in trying to get on topics which are on point or in vogue or intriguing or whatever. And today I think is an example of that. Today we're going to meander and we're going to think about the word psychedelic. And I'm really pleased. I've got two people, Tom, who's a good friend, an old friend, and Charles, who's now, I think that they call you Chuck, do they, Charles? If it's Chuck, we'll call you Chuck.
I go by either.
Oh,
there's a story behind that, but
you can be Chuck to us. But I've got as my sparring partners, I've got Tom and Chuck, and I'd like maybe them to just introduce themselves, say where they are, what they do, and then I'm going to get into the subject area, but maybe you could just introduce yourself, guys.
Chuck, Charles, you go.
Okay. So I'm a professor of pharmacology at Louisiana State University in New Orleans. I've been here since 2004.
have been studying psychedelics at the professional level since my postdoc at Vanderbilt University with Elaine Sanders Bush. So almost 25 years in the field. I started off in neuroscience, doing the neuroscience, gene expression, molecular genetic effects of psychedelics in the brain, developed fly models of serotonin neuropharmacology, and back around 2007, we discovered that psychedelics were potent anti-inflammatories in my laboratory. And so now about half of my research effort is outside the brain in the periphery, where we're focusing on models of asthma to look at SAR and drug development. A scientific co-founder of a company called Olusis, which is developing one of my drugs now for clinical use as an anti-inflammatory.
Excellent. And maybe Tom, you can just say who you are.
Yeah, I'm Tom Blackburn. I run a virtual development company, have done for quite a number of years. My introduction to psychedelics was back in the 60s with the Beatles and Lucy and the Sky Diamonds and all of those wonderful records. They were introduced to psychedelics in 1965 by a demon dentist, John Riley. And from that, they had a couple of years where basically, like a lot of psychedelic research, it went into a bad period with regard to its perception, with US governments, etc. But where I started was back in the 70s. So I've had nearly 4 or 5 decades involvement in developing selective 5-HT receptor antagonists, 2A, 2B, 2C, which went into the clinic. But one of the areas we wouldn't pursue because of things we'll discuss later is their potential in blocking a lot of the psychedelic-like compounds we used to use in our testing in animal studies. So I've had a long history, and I've looked at guys like Charles and his father David, basically bang the drum for psychedelic research over a long period of time. And I don't think it can be ignored anymore. And I think Charles will go on to discuss some very exciting findings that are starting to emerge.
So let's go back to the beginning, really. The word psychedelic. Just tell us, if I throw the word at you, Chuck, psychedelic, what do you think? Boom, here it comes.
Yeah, that's a charged word, I guess. The origin of it goes back to Humphrey Osmond back in the 1960s, who came up with a term for the type of drug like LSD, I guess, that combined psyche and delos, mind manifesting, to apply it towards drugs like LSD or psilocybin. But then all of these drugs sort of got co-opted under the term hallucinogen, and they were called hallucinogens primarily through the 1970s and the 80s and into the 90s. And then as research resurged a bit and clinical trials were happening, then sort of tried to distinguish drugs like psilocybin and LSD from other hallucinogens. And I think the field has settled on, again, trying to reclaim the term psychedelic for any kind of drug that modifies your perception that is primarily mediated through 5-HT2A receptor activation. So it's a dynamic history of the term.
Right, and so if I throw that word at you, Tom, what happens when I throw the word psychedelic at you?
Well, I think Charles basically covered it fairly succinctly. It's a mind altering drug, through consciousness, emotions, and auditory and perhaps visual experiences.
So we started in the 70s, we've wound our way all the way up to 21. So just say, maybe Charles, you could just give us a bit. So why are we having this conversation? Why is psychedelics all of a sudden becoming something worth talking about?
I know when I first got into the field, my father told me it would be the kiss of death for my career because nobody cared about psychedelics. At that point, they were a drug that, you know, if as Tom knows, if something came up in a drug screen and it was an activator of a 5-HT2A receptor, that was a program killer. So there was no medicinal value, no pharmacological value seen as developing something that activated the 5-HT2A receptor, and that was the dogma at the time. And it wasn't until late 90s, early 2000s when there were some private foundations and institutes that began to fund some human clinical work. And those slowly gathered some momentum and steam, sort of ultimately leading up to some very high profile studies at Johns Hopkins and Imperial College showing that they have these very long lasting antidepressant effects after just a single dose. And those clinical trials and studies have been followed up with additional clinical data showing some profound efficacy in substance abuse disorders, other psychiatric disorders. So psychedelics now, I think, are becoming regarded as sort of a real revolutionary new psychiatric medication. And there's really been no new class of psychiatric medication since the 90s, when SSRIs, I think, came out. So it really represents a paradigm shift in how to think about the treatment of several psychiatric disorders.
So Tom, is that how you would think about why are we bothering to talk about it in 21? Is it repositioning something that is old, or is it something that has caught up all of a sudden in our scientific world?
Yeah, I think in some ways it's transformative in what, you know, Charles was saying. You know, we've been 50 years or more, you know, trying to develop new drugs to treat mental disorders. You know, the SSRIs were the last, you know, drugs for treating depression. We have a serious need in the world for new treatments for depression. And the results, albeit, you know, small studies and not always hitting the primary, you know, endpoint, we are seeing some remarkable long acting drugs, both LSD, psilocybin, in depression. Now, you know, we've seen this with ketamine, ketamine, but what we're seeing with the tryptamine-like drugs is a very prolonged effect in the antidepressant properties. And, you know, this is something quite remarkable. And when you listen to the case studies, you know, the real world data, where patients are actually talking about coming out of major depression or treatment-resistant depression after years of in and out of healthcare clinics, hospitals, various antidepressant treatments, it appears to be something there. And, you know, what exactly that is, groups like, you know, Charles's group and others, you know, are trying to pinpoint it. But it may be, you know, and, you know, the more I think about this, it may be we're coming down to a sort of personalized medicine for the treatment, because what we're looking at, you know, perhaps with the 5-HT2A receptor, is that each of us have got populations of these receptors in our brain, in key areas of the brain associated with mood and emotion. And it may be that we can actually titrate, you know, the dose of these agents, these psychotropic agents, you know, for, you know, particular sorts of mental disorders. And as we know, there's a spectrum of, you know, disorders. So I think it's a very exciting time, but what is needed are good clinical studies, and to date we still don't have that many. I think there's something like, when I did a search of clinicaltrial.gov, we were looking at something like 63 studies with psilocybin and 119 studies with LSD, but these are all small studies over since the 1950s. So we're trying to bring this data in with modern day data to understand exactly what we're seeing in the clinic.
So if you, I mean, I've always thought that we were hampered by very crude animal models. I mean, is that what's, so LSD, great, or these drugs, great, been around a while, but before we can get into the clinic, Tom and Chuck, we got to get it through testing. I mean, are we still living, even with genomics, proteomics, and ebryomics, are we still living with swimming rats and things?
Yeah, that's one of the main issues for psychiatric drug discovery, and why many of the major pharma players have just gotten out of the business. They've just shut down their CNS divisions and repurposed the scientists, because it's difficult, and to model a psychiatric disorder is much more difficult than modeling something like a cardiovascular disorder. So that is one of the issues, and the National Institute of Mental Health here, the director of NIMH, has come down with new guidelines and recommendations over the last couple of years, saying that essentially we are not going to fund any more studies using forced swim tests, using elevated plus maze, things that really have no clear translational relevance to understanding human neurobiology and circuitry underlying these disorders. So the lack of really good, effective models that just don't meet predictive validity, but really a construct validity, that's been an issue. And that's one of the things that we've been doing in my laboratory, is to try to come up with some rodent models that actually will recapitulate what's seen in sort of the human demographic in the response of psilocybin and ketamine and some of these other drugs, to use them as discovery platforms.
I mean, Tom, I'm just thinking about the catching up of the way we approach science. Has it improved for psychedelics? Because if you take like antibodies now, we're much more used to jumping over animal modeling with antibodies. Now, Tom, can we jump over, you talked about the clinic, can we jump over into the clinic now with the psychedelics and just have a much better go at understanding it in man?
Yeah, I think what we've seen is a tremendous advance in the technology we have today to understand what we're perhaps are seeing in some of the animal models, particularly the non-human, primate models. But also Charles is, working on things like fruit fly, where, we can actually sort of show effects of these agonists and antagonize some of these behaviors. And we can, we're seeing this also, or starting to see it, perhaps in the clinic as well. So I think the technology, and particularly now we've got the crystalline structure of some of these receptors, we can actually see how they modulate the particular receptor of interest, in this case, the 5-HT2A. So I think there's been a tremendous advance since the 70s, 80s, and 90s, since I, was involved in these areas, to what we're seeing now. And we're looking at the microcircuits now within different brain areas. So we can actually image these in some cases and see how these areas inhibit or disinhibit each other. So we're getting a greater understanding at the molecular level, the microcircuitry level, and the imaging level in our basic understanding of, some of the animal models we've used in the past, or the ones that we're looking towards in the future.
So Chuck, thanks, Tom. So Chuck, I mean, where's psychedelic research going? We've already heard there's 2 people in stealth on this call, so they must be really busy. They're so busy they can't tell us what they're doing. But I mean, what are people doing now? Because we're starting to feel that it's just on point. It's something people are getting into. So what are people starting to do?
I think there are sort of two main sort of directions now a lot of people are going. You've got the clinical trial people developing psilocybin. And so their goal is to show that psilocybin is safe, it's effective to go into phase 3 clinical trials. But I think a lot of these biotech companies and things that are coming up now are, what's next? Is psilocybin the best drug? Can we improve upon that? Is there something with the pharmacokinetics or the off-target receptor side effects? Is it really necessary to have that 5 or 6 hour trip? So I think a lot of the companies that are springing up now are doing development to try to say what is the next step after psilocybin in terms of the treatment of psychiatric disorders, and could you further tailor it? Maybe psilocybin isn't the best for something that's not depression. Maybe if you're looking at bipolar, maybe another molecule will be better.
But surely, Chuck, we've done SARs for years on these.
Yeah.
Surely we've got cupboards full of SAR on psilocybin. So what's new? What's new?
A lot of it too. It's like the Wild West. It's a land grab. People going after intellectual property and indications. So it's also being seen as a business opportunity to try to identify what that is. And there's no real patent life to psilocybin. It's a natural compound. So you see Compass, what their IP revolves around is just a synthesis and an isomorph of psilocybin. So I think that there's not really much of a commercial opportunity in psilocybin by itself. So identifying something that could be potentially a better, more effective medication with some sort of intellectual property associated with that, then you've got a drug that a pharma company would be willing to invest the necessary resources to fund these phase 3 clinical trials. With all the work that's been done with psilocybin, it was all done by private foundation support over the last 15 years. Very slow going.
And I mean, Tom, has psychiatry moved forward enough that you can actually get a better readout of all these SARs that we must have buckets of molecules that are different? Has psychiatry moved forward enough that you can dissect the mind diseases and separate them enough to be able to start working on where they all fit in for use patterns, really?
The quick answer is no. The real answer is that you've got some very young, smart psychiatrists coming through now with all kinds of multidisciplinary, talents who are looking at, the structures of these receptors and seeing how the different psychotropic-like and psychedelic-like agents act at these, particular receptors. What we know from the SSRIs is that, you see a downregulation of this 5-HT2A receptor on chronic administration. But also, what we know from that is that the SSRIs will switch off, some of the adverse effects of the psychedelics. So it may be, that you'd be looking at sort of combination therapy, with some of these psychedelic agents with some existing drugs already out there.
But I mean, I go back to the point, back to Chuck, that immuno-oncology, having trispecifics, bispecifics, polypharmacy is just normal. I mean, is this new for CNS?
Not really. I think though, in the psychedelic space, there have been literally thousands of compounds already made. And you can just pick up Shulgin's book, TCAL and PCAL, and you can find the instructions on how to make several hundred of these. So there's a lot of SAR that's been out there, and it's kind of unusual because of the work that the Shulgins had done over the years. We have a lot of human data for the SAR as opposed to animal data, which is kind of the reverse of what you would see for a lot of this. So there's already a lot of that knowledge that's out there.
So if I asked you to do like a drug target product profile, what would be a clean psychedelic for the future?
But then you have to think that it's not just activation of the 5-HT2A receptor. It's how it's activating that 5-HT2A receptor. And what we know is like different psychedelics will recruit different effector pathways to the receptor, and those may influence the behavioral efficacy on the disease. So it could be that one psychedelic is activating the 5-HT2A receptor in a way that's better to treat depression, and say another one is activating it in a way that better treats anorexia. So I think a lot of the work now is not just sort of understanding how structures activate the receptor in general, but how are these different ligands activating the receptor. And I think there's been a tremendous investment in this by our own Defense Department. They awarded Brian Roth $26 million to address some of these issues on finding potentially better psychedelics that don't necessarily induce psychedelic behavior. Is there a way that we can activate the 5-HT2A receptor that doesn't produce a trip? So I know his group is going ahead with looking at those sorts of issues with this Defense Department funding.
So this is, I mean, classical, Tom, this is classical pharmacology, isn't it? You remove, it's basically something like a steroid. You've got, a psychedelic is like a steroid, a pluripotential, massive, it's a head sledgehammer, but Tom, they must be able to clean it up and come up with something that's streamlined and takes away, as you say, Chuck, the trip.
Yes.
Tom.
Yeah, and that is, exactly right. We know that, many of these receptors have pleiotropic effects, and it's trying to understand those pleiotropic effects and what parts of, say, the 5-HT2A, receptor will give you the trip and what part will give you the efficacy. Now, not only that, what we're seeing is with the 5-HT field, which there's now 14 plus receptors out there, is that we're seeing some allosteric, perhaps, modulation or some cooperativity between different 5-HT receptors. For example, we know a number of these agents have, 5-HT1A activity. So it's understanding how that interacts, whether that's efficacy or whether that's side effects, whether the 5-HT2C interacts, whether that's efficacy or whether that's side effects. So, really what we're trying to do now is to piece all of this information together in this jigsaw to try and understand, the good and the bad, between the, psychogenic agents.
I mean, Chuck, I mean, a mad thing. Is the trip important for actually the treatment?
Yeah, that's been a topic of debate for several years. And, is it really purely psychological or is it biological? And there was no real effective way to look at that. Like it's been proposed that maybe you could put somebody under anesthesia and see if psilocybin has an effect, but then you're putting somebody under an anesthetic that has effect on GABAergic systems and other systems that psychedelics are affecting. So that's significantly confounded.
Why would an anesthetic take the trip out?
If you put somebody under a general anesthesia, that will be affecting a lot of the GABAergic neurotransmission and the inhibitory interneurons of the brain, sort of the brakes of the system. And psychedelics target 5-HT2A receptors, which are expressed on GABAergic interneurons. And we've shown that psychedelics directly activate subpopulations of these in key brain regions. So there could be a significant confound. And on top of that, there have been some studies that have suggested that general anesthesia produces a rapid antidepressant effect. So it's really would be difficult to sort of tease it apart like that. And the approach that we've taken is, can we show that psilocybin has antidepressant effects in our animal models? And we've done that with rats. We've shown that there's a long-lasting antidepressant-like effect in rats. That is at least out to 6 weeks without any decrease. And ketamine is only effective for about 2 weeks. So that is about what's seen in humans. And then you could argue, does a rat have an existential sense of self? Does it have some trip where it realizes its space in life, its relationships, that can come to some of these understandings that maybe could have psychological consequences? And I guess you could argue that a rat does have some rudimentary system of this, but then we've also shown that a single treatment of psilocybin in fruit flies in the forced swim test, albeit, is as, if not more effective than chronic treatment with citalopram over a week to have an antidepressant-like effect. And could you make the same argument for a fruit fly, that it has some sort of sense of consciousness? So what we think is that at the core, that it's a biological effect, that it's doing something to synaptic plasticity in the cells, and without this biological effect, you're not going to get an antidepressant effect. But then some of our results have also shown that the environment that these rats have been placed in after the psilocybin will shape the outcome of the antidepressant effect. So when we put those together, we've come up with, at least in our rats, it looks like that the psilocybin initiates this synaptic plasticity and then this window of behavioral flexibility where they're subject to influence from the environment. So I think their subjective experiences, the integrative therapy that's after the session, I think it's all necessary to maximize the antidepressant effect.
I mean, just back to Tom. I mean, Tom, we talked a lot in last week about target engagement, and this feels like we've got the whole plethora of target engagement, and whether we're half engaging this target or fully engaging that target, plus a very unclear readout in the clinic. And that combination just feels like, my goodness, where do you go with it, Tom?
Well, I think there's two phase 3 clinical studies which are going to report in the next year or so, which will make it very clear. But I think what we've been somewhat held back by is, you're talking about 50 years of research, which there's anecdotal data. There's all kinds of new information coming through, with regard to trying to tease out what exactly these compounds are doing. And that's where we are today. We're teasing out, as a pharmacologist would do, as a behavioral pharmacologist would do, as a clinician would do. We're teasing out the good effects from the bad effects. Or, to answer your point earlier with regard to, ketamine. Ketamine is an anesthetic. So as you go up in dose, you get the anesthesia. Now, what we're dealing with today is microdosing, right? So microdosing has come into vogue with regard to how we're trying to understand. How can you get the sort of psychogenic-like effects with, say, 100 micrograms of LSD, but you can go down to 5 and 10 micrograms and see, the changes that you want to see. So, we're seeing dose-related effects through our studies, present-day studies, which we're trying to differentiate, the side effects from the efficacy.
I mean,
and that's pharmacology. That's where we've always been, and where we are today with this particular area.
You up the dose. Does it become a sedative? Do these psychedelics become sedatives?
No, they're not a sedative. They're actually they're stimulants. Some of them.
What do people die of, Chuck?
Taking other drugs, like heroin or overdose on other drugs.
So you don't get much death out of classical.
No, they're not. I think my father wrote something that there's been one reported case of death due to LSD, and that was because the police hogtied this person when they arrested him and suffocated him. So there's no known case of death induced by a classic serotonergic psychedelic that's been reported. So
you can take the dose right down, Tom, and you can split the effects then, can you, when you get to a really low dose?
Well, that's pharmacology. That's exactly what we've been trying to do across the years and decades, in taking the dose of the drug down where you can see the efficacy away from the side effects. And as you know, Charlie said, the effects with LSD, it's relatively safe so long as on your trip you don't fall off a bridge somewhere.
I mean, I'm still curious, both of you, why and what all these companies are doing that's new. Because I'm still struggling really to understand where the breakthroughs are going to be, other than repositioning it, because we've exhausted the other antidepressants, and the only hope we've got is some scaffolds that are around psychedelics. I mean, is that it?
I think a lot of the entities out there that have come up are looking at additional indications, and I think that's good. It's focused on depression and substance use disorder, especially for nicotine and alcohol over the last few years. But now with all these new entities and organizations coming up, they're able to look at additional indications. For example, OCD, looking at anorexia, other psychiatric disorders, bipolar, ADHD. So I think it's providing the resources and the infrastructure to see what else these psychedelics may be good for.
Yeah.
Good, Tom.
Yeah, Chad, just tell us about some of the really exciting things that are grabbing you today with regard to the research. And I think I'm referring to that Peter Hendricks at University, Alabama study. If this is right, this is quite remarkable.
Yeah, Peter is doing some really remarkable work. Like the clinical trials that they've been doing at Johns Hopkins and NYU and Imperial and the like, they have used very well screened participants, educated, people that they are not necessarily representative of the population at large, largely recruited from university populations, open to using psychedelics. Whereas Peter Hendricks, on the other hand, he is going and looking at the utility of using psilocybin to treat essentially homeless crack addicts. He is recruiting African-American, low socioeconomic, predominantly homeless drug abusers who think that psilocybin is a white man's drug. They'll do cocaine, yeah, no problem, but psilocybin, no, that's they won't even tell their friends about it because that's a white man thing. So they come in, they go through the therapy, and they've unblinded the first 10 patients, I think a few months ago, and the results are remarkable. That like 70% of these patients either were abstinent, they stopped and quit. A number of them got assistance from social services to get themselves out of where they were. There was some concern that they would just go right back into the hood and start using again, but that turned out to not necessarily be the case for most of them. So it's really exciting to me because it's the only kind of real world study that's out there looking at a population that's been neglected and overlooked that presents a lot of different challenges. socioeconomic. They're going back to the environment that they came from. And it's really remarkable what he's shown. I think that's more representative of real world use.
So, I mean, that's a brilliant, I mean, we're going to move into the questions in a minute, but I mean, what I'm hearing here, and maybe I can ask Tom next, is that the future feels very exciting with the psychedelic world, that really we haven't looked enough across the clinical, if you like, landscape. And what people are now doing is looking at all the different types of people, all the different indications, and seeing that there's clearly a place for some of these psychedelic. Is that the future? The future is a good one if you work in psychedelics.
Well, you know, again, yes and no. You know, the legislation and regulatory issues, you know, are something that, you know, needs to be looked at and is being looked at. You know, we're seeing the FDA, you know, fast tracking, you know, some of these, you know, particular studies. And we've already seen it with, you know, in the cannabis area, you know, and Chris, you know, can talk about this later. You know, we're seeing clinics now in the US, you know, ketamine clinics, we're seeing LSD clinics, we're seeing psilocybin clinics, where, you know, patients are going there, you know, to be treated for their, you know, severe depression. And, you know, how that evolves, you know, we've already heard of, you know, investment from, you know, the Canadian government in, you know, some of these areas. How that evolves will give us more data, I think, on, you know, the success or, you know, failure of this area. But from what I'm seeing, you know, I'm seeing a greater understanding, you know, with regard, you know, to this particular area of, you know, therapeutic intervention.
Well, that is fantastic, guys. I'm going to bring in the chat because we're now coming up to 50 minutes. Thank you both so much, and let's start bringing in the audience. And we've got several questions. Graham, do you want to get a couple of questions?
Unsurprisingly, we have loads of questions, so we're going to start with Sue Jackson and then go on to Shahar afterwards.
Thanks, Graham. Thank you, Tom, and thank you, Charles. We've focused mostly on depression so far. How about schizophrenia? Would you see that as in much the future looking much the same or different? I spent I was at Imperial in the mid-60s, so that was my encounter with psychedelics. But subsequently, I spent a few years working on a GlyT1 for schizophrenia, which didn't make it. So I'm quite interested to know, in terms of psychiatric strategy, depression versus schizophrenia, similar or different?
Well, I think if you look at the most successful antipsychotics today, you're seeing a combination of D2 and 5-HT2A, you know, antagonism. So there's that 5-HT2A receptor. But I think Charles could answer this a bit more with some of, you know, the more recent studies, you know, with regard to, you know, this particular area. You know, particularly with the auditory and the hallucinogenic type aspects of, you know, schizophrenics.
Tom.
Yeah, there's I know that the NIH here has spent a lot of money over the last several years devoted to trying to identify the etiology of schizophrenia, because I think they've taken a step back, saying, all right, we've got these 5-HT2A D2 combination blockers. We can treat the positive symptoms, but we're not really addressing the negative symptoms, even with the 5-HT2A blockade in there. So they're taking a step back, and they've done the genomes and sequencing and trying to do these GWAS studies. And I think what they're understanding now is that there's not 1, 2, 3, or 10 genes that are involved in schizophrenia, but there are literally hundreds of genes with a low effect size, and that schizophrenia is not really a one size fits all. So I think there's been a lot of understanding at the circuitry level, the basic level, on trying to better understand schizophrenia. but there still haven't been really any new medication developments that have come out of that yet. And I'm not sure yet that there will be, because there's not a single or 2 or 3 druggable targets that are you're going to be able to go in to really hit and enhance. Maybe I think Herb Meltzer was saying, well, what antagonizing the 5-HT7 receptor, adding that in may have some greater efficacy. So there have been some new antipsychotics that are antagonizing, I think, the 5-HT7 receptor and some other different combinations. But at its core, it's still D2 and 2A blockade, and that hasn't changed. And I don't really see that really changing much over the next few years.
Thank you.
Great. Okay, we've got another 5 questions to get through, so we're going to go for Shahar next.
Sure. So a couple of years ago, I was involved with a family member who had cluster headaches, and there is a large patient population that are dosing themselves, microdosing with psilocybin, and that's a much easier, I think, target, because it's less on the psych side and it's more on the bio side. It's a known, the biology there is, and it's not understood, but it's there is much easier to test for it. And I've seen less work on it, and I'm trying to understand why is that, A, and B, what do you guys think of this? Is this some kind of a side effect, or is this something, is there something real there? For my family member, it worked. His cluster headaches, which is a really horrible disease, he has less occurrence of that, much weaker. So I'm trying to understand the difference here, because a lot of the CNS diseases are much more, there's no good animal models for those.
Yeah.
And there are good animal models. Yep.
Yeah, I think that's almost a vascular disease as opposed to a CNS disease. And Emmanuel Schindler at Yale is doing clinical trials now with psychedelics and migraine headaches. And she has found, interestingly, that the therapeutic efficacy of psychedelics to treat migraine doesn't really depend upon the individual having a peak ego dissolving experience, such has been correlated with the antidepressant effects. So at that point, you don't know, as to getting back to Tom's earlier point, it may not just be the 5-HT2A receptor. It's interacting with 5-HT1A, with other receptors. It could be the 1D receptor. LSD is an activator of that. But I think it's interesting that, unlike for depression, the peak experience is not correlated with the therapeutic efficacy. And so they're I think primarily doing clinical trials, but it's the basic and fundamental biology is not known.
Yeah, I think
they're all microdosing. Yes. Sorry, Tom, go ahead.
Yeah, they are microdosing, and the interesting thing about I took a compound into the clinic with 5-HT2A B/C activity, back in the 80s, and it had some marginal effects in migraine, but we never really put enough time, money, and effort into those studies. But there is a good pharmacology around the cerebral vasculature, with regard to these 5-HT subtypes, particularly the 2A and the 1D, and certainly sumatriptan, which has been used in migraine, has this 5-HT1D like activity.
Great. Okay, we've got Anna Stenstam, please.
Yes, great. Well, I'll make you happy by combining all my comments in one, I think.
Good
Hey,
stuff.
saving time. Okay, so from my perspective, it was really interesting to hear this with, of course, do fruit flies have a sense of self, and do they get trips? I mean, I think I will go to bed thinking about that. But the question that I have is related to that, because if these are so complex things, and still as scientists we go discussing, one receptor, this is the target, this is the mechanism, but still we're also acknowledging that this is probably much more complex and personalized and all this. And then, since I also work with food and cosmetics and other things where we can go to the market with things that we don't really understand, but we see that it has an effect, right? Like, you know, you eat certain foods, you don't always know exactly what does what, but you know that if you eat a little bit of everything, you will be doing better. So I know pharma, I am also working there, so I know it's more regulated, but I have to ask, is there any discussion around with FDA or whoever about, you know, making studies in a different way because this is so different? Or is it exactly the same as, you know, with all the other drugs and we have the phases, blah, 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 blah. Or is there any possibility to have such a discussion to make, I don't know, super control, functional MRI studies to get the mechanism, and then you move on to a bigger population, but super controlled? And I don't know, how dangerous is it to take that, to jump some things and be a little bit more iterative?
Yes, I was at a conference last week that was organized by the FDA, specifically to talk about clinical issues about the development of psychedelics and how the FDA can be involved in doing that. And several of the higher up people at the FDA spoke, and they were all very enthusiastic about developing psychedelics as a novel therapy. And the administrator who gave the presentation said that this might be surprising, but most of the drugs that are FDA approved, we don't know how they work.
Yes.
So you just have to show, and he read an example of a label that said, well, the exact mechanism is not known. However, there is a theory that, or something like that. So all you have to do for FDA approval is show that a drug is safe and that it's efficacious. You do not have to have one inkling of how it works. I think the knowledge of mechanism really gets towards the drug development and discovery and how to identify better drugs. But for FDA approval, just safety and efficacy. So a large part of that discussion then after that pivoted towards, well, how do you show that it's efficacious? Because with this class of drug, it presents a very unique challenge because you cannot really do a placebo control. Because if somebody gets a high dose of a psychedelic, they're going to know it. And the person, the investigator, is going to know it. And then there's also a nocebo effect. So a lot of the discussion was really, how do you prove the efficacy of the drug itself when you cannot properly do a placebo-controlled arm? And then there was discussion of, well, what about using a benzodiazepine or using niacin, but none of those produce the subjective experience of a psychedelic. And so from the FDA's perspective, that's the most challenging aspect of the development of psychedelics for clinical use is the efficacy.
But that's the same with acupuncture and things like that. I mean, you know if you have a needle in you or not. So, I mean, all right. Okay, but it's better than I thought. More discussion than I thought. Thank you.
Okay, very good. We got Chris Day next. Hi, Chris.
Fantastic to have you here, Chris.
Yeah.
Chris, Chris, can you just tell us who you are and what you do?
And how I know you.
No, we don't want to hear that. We've already heard. And that's private. Let's hear Chris.
No, I spend most of my time outside of the science, but really talking about the business angles of cannabis and exploring ethical and responsible business developments for legal cannabis markets. What's interesting to me about the psychedelic space, though, is that many of my colleagues who have been on sort of the front edge of the cannabis landscape are now also shifting into psychedelics because, to your discussion around the FDA and research, right? We've got multiple issues. I don't think anybody debates that there is medicinal value within psychedelics, within cannabis, but that doesn't actually matter when you're a politician or you're a bureaucrat in the FDA. What matters there is whether or not you're going to get votes. So we're we're balancing the political will to allow studies to come forward with the very real need elsewhere. So we talk about all these studies and human trials, and I can show you many neighborhoods in Houston, Texas, where there's plenty of people doing human trials on their own that will tell you all of these things that we've been talking about work, right? So I think the most interesting thing I heard so far was the reference to the work with Peter Hendricks. And what I'm wondering is, like, when you're working on these projects, how are we communicating the long-term perspective value, even if it's not completely figured out yet, the long-term perspective value to the general population, because we've got to create a groundswell of desire there for the results. Otherwise, all of this theory will just take another 40, 50, 60 years to actually ever get into the marketplace in a way that's perceived as legal. I mean, here, at least in Denver, we have decriminalized psilocybin, right? Which just encourages more self-treatment rather than actual focused treatment. So I'm curious to know how, as these studies are going on, you know, you're communicating it in bite-sized, consumable, you know, and understandable ways, other than receptors, because I dig it, I love it, but it's not helping the greater cause to really perpetuate the discussion.
I love the 60s language. Dig it, trips. My kind of place we're in. I don't know whether Tom or Chuck, you've got any comments from Chris's.
I think at an individual investigator level, like with Peter and some of the other work, there's not really a direct outlet to the public to understand this. Like, the public will see that, oh, it's Johns Hopkins, or there's a new psychedelic center at Harvard. It must be legitimate. But exactly the type of research and therapies that are going on, I think to date, it's either social media of a lot of press releases, but then there's a lot of misinformation in social media. And I think probably the most effective platform to date to get these results to the general public has been Michael Pollan's book on how to change your mind. That really brought a lot of what has been going on, distilled it down into one text that people can read and go, oh, wow, I didn't know that psychedelics could do this. In the 60 Minutes episode that Roland Griffiths was on, I think those sorts of things are few and far between, and we could have more of those. The scientific community at large has not been successful as usual in connecting with the general public.
Yeah, you know, Chris, you know, to answer, you know, your point, it takes me back to when Glaxo were promoting their anti-migraine compound sumatriptan, and everything obviously is based on the clinical trial, you know, results. But there was a drip feed that went on for a number of years, whether it be in women's magazines, TV, you know, slots, where that information, you know, was out there, you know, with regard, you know, before social media, was out there in newspaper articles and whatever. And I think, you know, the cannabis area to me is, you know, perhaps 10 years ahead of, you know, where, you know, the psychedelic area is now in evolving. And that may accelerate quite quickly, you know, with respect to social media. You know, once the word is out there, Chris, and that, you know this well, once the word is out there on the street that, you know, these compounds are effective in, you know, migraine cluster headaches, in treatment-resistant depression, in obesity, or whatever you'd like to say. Once that word is out there, it is so quickly taken up then by, you know, the media. And patient groups, we've seen it so many times, are advocates, and they lead the way with the politicians. So if the politicians need your vote, they've got to listen to these patient groups, and they're the ones with the power
in theory. So
Theory.
Yeah. One thing I will just tell you observationally, I was talking to some folks doing research in Israel yesterday, and in the psychedelic space. I do have hope that actually this discussion will have greater velocity in the broader community, because it does not face quite the same level of propagandic vitriol that cannabis has faced in being tied into, say, Mexican drug cartels or Pablo Escobar in the 70s, right? And so there's ingrained psychological trauma culturally around cannabis that is less intense with the psychedelics discussion. So I do think there may be a leapfrog effect, plus all the work that has already been done in cannabis to overcome some of those objections. Those walls are slightly lower than they used to be now. So I think there's a greater opportunity. I would just encourage you to continue to communicate these types of benefits out more broadly, because this discussion is wonderful. But if nobody knows it's happening, it changes nothing except scientific theory as opposed to the greater good of the community.
Mm-hmm.
Fantastic, Graham. How are we doing for questions?
Okay, well, we've got a few more questions, and I think we've got to get through them in the next kind of 7 minutes. So we're going to go with Nick Moore next. Nick, you got your question there?
Yeah. So it's a couple of points. First of all, on the animal models, my background, I'm in drug discovery for many, many years, behavioral pharmacologist. I don't actually think there are animal models of CNS disease. You can't model the psychiatric symptoms in the animal. What you can is use it as a mechanistic assay to show that interacting with a particular mechanism will change the behavior in an animal in a certain way. And because of that, the animal models tend to only have predictive validity. So some people say, you know, you can have similarities, but I believe that they have predictive validity. So anxiolytics work in that model because benzodiazepines work. SSRIs and antidepressants work in a model because SSRIs work in that model, but it's not because the model's a model of that disease. And I think we should avoid, you know, bad-mouthing the animals by doing that. But my other point then extends that into the clinical trials. In depression, things are much more complicated in depression than, you know, one particular symptom, and the trials that we use to measure depression maybe just aren't fit for purpose when it comes to using new entities that aren't SSRIs, aren't standard antidepressants, or the same in, you know, anxiolytics. If they aren't benzodiazepines, then they don't show up in a trial that's designed to show benzodiazepine-like efficacy. I'd just be interested in your comments on that.
Yeah, like for your first point, yeah, that's very true. And NIH here has recently has this research domain criteria now that they no longer say think of a disease as a model. You know, you can propose an animal model by, you know, don't really call it an animal model. It's an experimental system to study some aspect of hippocampal dysfunction, which is relevant to understanding a mechanism of learning and memory in dementia or something like that. So they've really pivoted over the last, I would say, 5 to 10 years into studying and developing animals to study aspects of disease that are shared amongst other diseases, and they're calling it the research domain criteria. So that's I think been received fairly well, and a lot of investigators are going that way. And I myself am catching myself very frequently correcting myself. Oh, it's not an animal model. It's an experimental system to study X. And for the second point, yes, and I think that's shown up in some of the clinical trials where you have Compass is looking at psilocybin for treatment-resistant depression, whereas USONA is looking at psilocybin for major depression, and you have all these subtypes. And I think Johns Hopkins did depression associated with cancer and NYU did anxiety associated with cancer, but there's no DSM for cancer associated depression and anxiety. So they couldn't do that. So there's several different facets of psychiatric disorders that are all disjointed at this point. So unless,
and I agree with regard to that second point. I think, the DSM, criteria is outdated. And, one of the good things what I'm seeing is that, the regulatory authorities like the FDA, MHRA, European, are actually willing to sit down for the first time and actually discuss the design of the studies. So I think, with these alternative designs, I think we may be seeing inroads in the future, with regard to, better clinical studies. And certainly, when we talk about subjective behavior in animals, this is what we're actually diagnosing in humans. It's, the DSM is a subjective behavior, criteria. So, we've got one bad thing, perhaps against another not very precise tool. In fact, a very blunt tool in understanding what we're trying to do.
Exactly. How are we doing for time, Graham?
We've got two more questions. We're going to get through these questions. Ian Reynolds, if you go first.
Okay, thanks. So I really enjoyed the discussion this afternoon. So thanks everybody for that. Still on the topic of clinical trials, what strikes me about this field is that the ratio of anecdote to data is extraordinary. Right. I mean, with all due respect to the late Sasha Shulgin, I mean, he relayed a lot of anecdotes, but very little actual data in terms of human clinical effects. And it's like that everywhere. So I guess the question would be, do you have a sense of where the best opportunities are to actually get hard clinical data to support the use of any of the psychedelic compounds? Is it going to be in depression? Because I mean, depression clinical trials have always been so difficult. Is it someplace else? Where's the data actually going to come from first, do you think?
I think it will be the anti-inflammatory effects of psychedelics, because that you've got clearly, if you have inflamed tissue or organ and a psychedelic is taken at a sub-behavioral dose, you can do an accurate placebo-controlled study and you can have a defined physiological outcome. And that's what we've been developing in my laboratory along with Elucis is the anti-inflammatory side of things. And so I think with those we can do traditional drug discovery, clinical trials, similar to any other drug with proper controls. I think it's when you get to the psychiatric side of things, that's when it becomes difficult.
Yeah, whoa.
Okay, go on. Sorry, I was going to say last but not least, we're going to go for Charlie Oliver.
Hey. Hi.
Hi, Charlie. How are you?
Good. Thank you. How are you guys? I wasn't really expecting to be on camera, but here we go. So thank you so much for this conversation. It's awesome. Here's the thing. I've been in a lot of these events. I work in tech. I'm the CEO of Tech 2025. And I have been hearing, because we're covering this in the coming months and with clients, believe it or not, I've been hearing two different spectrums in this conversation, ongoing conversation. And one is you're at one end of the spectrum, right? Which is the clinical research, which is really important. And there's a lot that needs to be done. The regulatory issues, that. And then the other end of it is the sort of, I'll call it the tech bro thing, where, the technologies that are being developed that don't in any way, they're not meant to replace the actual experience of taking anything, it's just, I think a lot of people will be introduced to psychedelics through technologies because it's an easier way. And I'm not saying again that it replicates the experience exactly, but the immersive technologies like VR, AR, or whatever apps. And my question is, companies are going to be ground zero for a lot of the mental issues that are coming. How do you see companies embracing, if at all, psychedelic therapies, whether through technologies or other ways in the next 5 years as a part of their mental health and wellness programs? Taking into consideration the technological aspect, because there are companies now, I have clients who are asking me about it. They know they can't go the drug route, but
Yeah, one of the interesting things about where we are today is, we've got this sort of aging, stay young, stay relevant sort of philosophy, which is, particularly evolving, with a lot of these investment groups, the sort of A-type, professionals who, whether that be in the financial community or, whether it's, elsewhere, where basically, they're looking, we've seen it with modafinil, we've seen it with Adderall and one or two other compounds, where people want to be smarter, more cognitive about, changes. So, that's one aspect which may drive it, that, there's a health and well-being aspect to this. But, my sense of the whole area is, we've been waiting, when we were waiting on the first, COVID, vaccine. I think once we start to see some real world data in any of the sort of areas that we've been discussing, I think, you'll see this sort of domino effect where, more people will become interested in this area. And at the moment, we're waiting on some key studies, whether that be cluster headaches, depression, etc. So I think, we'll see some real world data, in the next 12 months, which may certainly accelerate, this whole therapeutic field very quickly.
Thank you very much, Gerd.
Charles, do you have any comments on that? Carry on, Tony.
No, I was going to say, I think we're at the end of our question time.
Okay.
What are we going to do next? And before we do something next, firstly, let me thank Chuck and Tom in our traditional way. Super duper.
Thank you.
Can I just say super duper? We're really up for this next time. Perhaps you can come back, because we'd like to broaden the discussion again further, and maybe get some people who think of psychedelics more in a spiritual way, because I'd love to see that conversation. Really looking forward to that. Graham, what's the score on the door?
So